On this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
Welcome to Life Science Success Podcast. For those of you who don't know me, my name is Don, and I'm a consultant in life sciences. And if you haven't taken the opportunity yet to join me on my weekly series, I've been running this weekly series called Thoughts at Scale. And the Thoughts at Scale is really meant to dive into key topics that help organizations with regards to scaling in life sciences. And so as you look across the different verticals that are here, we primarily focus in on the consulting side on helping companies with strategy, people, process, and technology. That's normally the way that I explain it real quickly for most organizations. And the Thoughts at Scale is really meant to give you kind of introductory lessons in terms of things that would prevent companies from scaling. And some of the key examples that we've covered already would be things like roles and responsibilities. We've talked a little bit about strategy and vision. We've also covered the process optimization side of things. And this week, on tomorrow's session at noon Eastern time, we're covering metrics. And so this will be a great conversation for organizations that are looking to develop metrics that tie to their strategy that then lead to overall processes that support your organization scaling. And so, very important for organizations to have as you think about, scaling in the broader term. So with that, let me bring in our guest. In this episode of Life Science Success Podcast, we're honored to have Dr. Alan Trounson. He's the CEO of Cartherics, a company on the forefront of redefining cancer treatment through immunotherapy. So with that, welcome to Life Science Success.
Hi, Don.
Yeah, thanks a lot for being here. I really appreciate it. And would you just take a moment and tell the listeners a little bit about yourself?
Yeah, I'm a reproductive scientist. I graduated in wool and pastoral sciences, which was really wool technology. So I was interested in agriculture as a kid growing up and going to university. So I spent my time with animals for a lot of my time. But I ended up doing a PhD on sheep embryology. And that introduced me to Carl Wood, who was doing human IVF. He offered me a job to go to Melbourne to help him do human IVF. But I went to Cambridge instead, worked at the Agricultural Research Council unit as a postdoc there. That's where they were doing some absolutely incredible work. For example, Ian Wilmot was there. So he and I were friends, and Ian's just died, and Jose Sibeli and I have just written an obituary about Ian in Nature Biotechnology. But we had, it was an inspiring time. I spent 3 years there, and they wanted me to stay on, but we came back to Australia, and I got involved in the human IVF thing at the time. I thought, well, we were doing it in animals. I can't see really why it wouldn't work in the human. And Carl gave me, he allowed me to do what I wanted for a period of time, for a year or two. And we actually translated what we're doing in animals over to the human, and it worked. And so that's the basis of human IVF. And since Edwards and Steptoe had to move from where they were in Oldham to Bourne Hall in Cambridge, they were off the map there for a couple of years, 2 or 3 years. So we were actually training everybody in the world in human IVF. So it was a very incredibly interesting time. So in the 90s, I decided that we should try and develop human embryonic stem cells. And that worked also. I worked with some colleagues in Singapore, and that worked. And we had the cells when Jamie Thompson actually sent us the paper, or the journal sent us Jamie Thompson's paper, I should say. And we already had the cells in the lab, but we knew that it would work, so we gave him the thumbs up for his paper, but published quickly behind him. But having made embryonic stem cells, these pluripotent stem cells that are they're immortal and can develop into any cell of the body. Suddenly I realized there was a power there that could be harnessed for human medicine. So the Australian government gave me a large grant to form the Australian Stem Cell Centre here. But I went back to the Monash University to keep the research going because I thought it was too early to sort of do those things. But eventually, Bob Klein and Arnold Schwarzenegger talked me into going into California to head up the California Institute for Regenerative Medicine. And they had $3 billion of Californian money that they received in the usual processes there, bond money. And so I spent 7 years at CERN in California, San Francisco, igniting a revolution on stem cells, which was magic. Suddenly having $3 billion for a scientist is rare. But it gave me access to so many fantastic scientists, particularly in California, across the world, because everyone was really interested in what was going on there. And so that when I finished up after doing 7 years, I thought that was enough and come back to Australia. I set up Cartherics. I didn't want to go back to the university, so I thought I'd try doing an immunotherapy, which I thought was the best thing that we were funding while I was at CERN, which is a chimeric antigen receptor technology, the CAR technology. And there wasn't anything much happening in Australia, so I thought there was an opportunity, and so we started CAR together with some colleagues, and that's a potted history of an opportunist, Don, if you like.
No, that's perfect. And I always, sort of enjoy these journeys that people take because, oftentimes, you could start in one place and thinking that your life is going to lead you somewhere else. Did you ever envision, though, earlier on in your career, did you ever envision that you would be the CEO of a company like Cartherics?
No, never. When I was an academic, I relished the time in academia doing research and doing things which I wanted to do, not what other people wanted me to do. And I thought it was very special. And having had some success, at least measured in terms of recognition, the IVF and the stem cells received there was a lot of debate about that work, about whether they should happen, particularly in Australia and some other places, and in the US as well. I've received a lot of recognition, some critique, of course, from some sectors, but it kind of worked. And for IVF, there's, I don't know how many, 15 million babies, 20 million babies around the world. If it hadn't been me, it might have been somebody else. But we were there and set it up in the beginning, and it feels very good. And I feel the same about now what's happening with stem cells. And then I thought a biotech company, heading up a biotech company. What I needed to do was really have some good people with me, because you really do need good business people. It's a tricky area to be in a biotech company. Most of them go aground, I think, as you realise. But we've kept afloat with really smart people beside me. So, I think if you've got really good people and you've still got a strong vision, you'll succeed. But sometimes it's not easy.
Yeah, it's funny because I've been working with a company just recently that had a great marketing vision, but it's that element of tying it back to how are you actually going to deliver this thing. That I remember the first time that I'd asked that question and the long pause that came afterwards. Because the person was trying to think through, like, how do I take this now and tell people about the steps that have to happen, et cetera. And so, yeah, it's quite an interesting thing to in organizations that don't have that person, that can be the integrator, that can think through how do we turn this really into a business as well. And then you're dealing with whenever I think about it, primarily with CAR T therapies, you're dealing with something that has a relatively short shelf life once it's all put together as well. And so, maybe we should just talk a little bit about what Cartherics is doing in terms of, what is your mission and vision, and what is it that you're bringing about? And then we could talk more about the therapy itself.
Okay. So Cartherics was set up with me as a stem cell biologist, and really without any good, strong immunology. So I did get a tremendous immunologist came out of Monash University to join me. We'd worked together before, Richard Boyd, and I put together a team including Ian Nisbet, who'd been at Millennium and other places in the US and Australia, and in the business sector delivering on these. So with that group of people, we decided that, what we wanted to do was try and bring the CAR T work from individual patients, high cost individual patient technology, where you had to take the patient's own cell and genetically engineer them, turn it into an off-the-shelf product where you could treat hundreds or maybe thousands of people with the same product. So then we thought, well, there's so many people in the blood cancer area, particularly in the CAR T area, working on CD19, which is a B cell marker. We didn't want to get into blood cancers because there was so much competition there. We decided to focus on the solid tumours, which is where most cancers are. And these are the difficult areas. So we took take on the difficult area. We focus Cartherics on solid tumours, like ovarian and gastric or pancreatic, lung, liver, etc. And we wanted to utilise sort of rare, what I call broadly histocompatible umbilical cord blood samples, which we converted to induced pluripotential stem cells. These are closely or nearly equivalent to embryonic stem cells. They're immortal and they can develop into any cell of the body. So it gave us the power of being able to multiply those cells incessantly, and so therefore ideal for gene editing. And we could differentiate them into immune killer cells. So we wanted to differentiate them into T cells, natural killer cells, macrophages, all these kind of things. And that they have this off-the-shelf product, it should be at low cost and high patient coverage, so it would address the problem that it's not available. CAR T is not available to everybody because of the high costs. So our vision is to ensure cancer is a manageable condition, not a and be available to everyone. So that's how we set up the company.
Very good. And then how do you stand out? There are a lot of companies I feel like that are working on NK, to your point, NK therapies, CAR T therapies, but how does Cartherics stand out, versus the competition?
Right. So we're using killer cells of the immune system, and these immune cells have been evolved over millions of years to protect us against infection and dangerous cell changes, especially cancer. However, the normal frequency of such cancer-specific killer cells in the blood is very low, and they're inhibited from killing the tumour cells because of protective molecular shields that exist within the tumour itself. They're within the tumour microenvironment. And so they're not really able to deal with the cancer. There are few of them, and they're being turned off. So recent advances in genetic engineering enable the technical insertion of immune cell genome of cancer-seeking and destroying molecules like CARs. They're attached to the surface of the cells, and these CARs recognise specific cancers, so you can put multiple CARs in there to stop them escaping the killing part. And if you multiply these up, and we intend to use this as an off-the-shelf technology, producing very large numbers of these cells, but they're also designed to overcome the immune inhibitors of the solid tumours, and also to recruit the patient's own immune system to help eradicate the cancer. So we're using them not only to kill, but to dampen that inhibition that's existing there. It's a very strong inhibition. Kind of macrophages that live in there don't do anything. They actually protect the tumour. You need to convert them to the cytolytic type of macrophage. You need to bring the T cells back into action and killer cells and so on. And so we're actually modifying our NK cells to not only kill, but to make that environment suitable for the patient's own cells to get in there and do the job as well. So then you got, you're bringing in multiple armies into this job. And we strongly believe this will open cancer to immune management. So that's how we think we differ. We want to disrupt that tumour microenvironment, open it up, make it more like a blood cancer or an infection, and then get the immune system to deal with it because it's a powerful tool. And we'll enhance that by putting in multiple cells with lots of cars on them, right?
Yep. And what sort of phase are the things that you're working on right now? How much longer before we start to see human trials, you think?
Well, we expect the human trials to start, our human trials on this to start in 2025. We're actually got a CAR T, which is an autologous product out there to test, and that's testing our CAR target, which is called TAG72, with some gene knockouts of the principal inhibitors, and we wanted to check that out. Clinical trial, we expect next year or probably early 25, but our off-the-shelf product should be in the clinical trial in 25. So we're currently in discussions with the FDA. We're applying for a pre-IND at the moment. We've had a discussion with them. We expect that this will allow us to get to an IND in 25, and we'll have the cells ready in 25 to start treating patients. We're going to focus on ovarian cancer in the beginning because it's such a dreadful disease. Patients who have ovarian cancer really, if you don't survive, if it comes back after the initial surgery and chemo, you've got very little chance of survival currently. So we thought that let's start with that disease. It's dreadful, and it's something that we'll set our focus on, and then expand the type of cancers thereafter.
Yeah, and I'm not familiar with the patient journey today in terms of ovarian cancer, but in the 70s, my mom passed away of ovarian cancer. It's one of the reasons, why I care so much about life sciences and seeing these things advance. And so, even though it's awful to lose a loved one, at the same time, I sort of, say just like cancer is a good fighter. It makes a really good series of enemies as well, because a lot of us that were impacted by it have said, you know what, we're just going to sort of dedicate our lives to making sure that people are successful, creating ways to fight this thing so that we don't lose more good people in the world, right? So, yeah.
Yeah. So my only feeling there is that there's a tendency to sort of just try things rather than sort of go deep enough to address the real basic issues that you need to. If you don't, in ovarian cancer, you don't open it up to genuine attack from our immune system, it doesn't really help that much, to use monoclonal antibodies and other things. It just doesn't get there. It just doesn't do it enough. And so we don't think that our first product will be ideal, but it'll be the beginning. If there's any sign of that, we'll keep adding to those blocking those inhibitors, and we'll bring in, more of these, the accessory strategies to really keep opening up that tumor. I think it's well exemplified in using the checkpoint inhibitors, because that allows a person's own immune system to function, and it works really well in some patients, but not all, and unfortunately very few in, say, ovarian cancer or some of these solid tumours. But if it does, it works really well. And that's just really engaging the patient's own immune system. So you see, if we can open it up, engage the patient's own immune system as well, I think we'll make a difference. And that's really this is really what's setting us aside from everybody else, I hope.
Very good. And then can you just talk a little bit more about the role that gene editing is playing in your therapies as well?
Yeah, gene edits allow to insert CARs into known safe harbor sites in the genome and to disrupt genes known to inhibit immune killing, so they can extend the life of the CAR-killing cells, the CAR cells, and enable immune memory cells to be established and prevent recurrence of regional cancer and other related cancers. So the gene edits are really crucial and can only really be done if you've got a cell like an iPS cell. Because what you really need to do is to be able to show the gene edit is accurate, is in the place that it intends, and hasn't disrupted something else. The worst option would be if you disrupted an oncogene and you created, a cancer-forming cell, in what you're giving the patient. So if you can clonally derive them, you can actually and deep sequence that clone, the cells derived from that clone. You can see exactly where the edit is, and you can see that there's nothing around in any of the cancer genes, the oncogenes, and nothing in the functional genes. So it's the same as the original iPS cell. You can't really do that if you start with adult cells because you can't multiply them like that. You can't clonally derive them. So we end up with a product which is much more like a drug product, a pharmaceutical product, which is what the pharmaceutical companies are looking for, a defined product. And also the regulators prefer to have something that's defined because then they can say, well, that's that. And it's not a whole mixture of things, which is what CAR-T is, a mixture of things. Cells have got a mixture of things happen to them. So the gene editing, I think, is absolutely crucial. And it allows you then to sort of scientifically develop what you want very accurately. So you can sort of knock it into a certain place, and then you can maybe put multiples in there or choose multiple sites for your CARs, so you can have multiple CARs. And you can do knockouts of specific genes that you want. And so long as those knockouts don't prevent your cell differentiating into your T cell or your natural killer cell or your macrophage, it'll be fine. But you need to test that out. The knockout needs to be consistent with you producing your product. So this is a very, very powerful addition to what we didn't really have in the past. And I think it allows us to design products that are very specified in what we want them to do.
Very good. In terms of partnerships and collaborations, are you currently engaged in partnerships and collaborations at Cartherics?
Yeah, we're working with some really innovative scientists and companies, and because we're out there on the edge a bit, sort of looking to do things differently, these are in kind of related technologies, but not what you'd think they were. So we do have good partnership with Trounson, a company in Korea. We have some very interesting other relationships with Chinese and Malaysian and Australian groups and Korean groups. So these are sort of people who are doing things a bit differently, and we think that that's what we want. That's what we want to draw in, and we keep talking to people who have the intellectual property for some of the things that are not being used in the way we want, but we think that if we can haul them in to work with us, either in co-development or in some way sort of helping us get to the end point, but also helping them, this sort of works. So we're a bit different to other companies in that regard, I think. So our colleague, we came out of a university, so that's also helpful. We're closely related to both Monash University, but also Melbourne University, but also the Cancer Institute. So there's a sort of deep relationship. Then we have federal government grants. So because we're scientists that come from the academic side, it helps when you put in grants
Right.
as a company, because often you've got to have an academic sort of reviewing your grants. Um, and so we're deeply embedded in innovation, and we hope that is going to give us the tools to do what we want to do, to manage this cancer. And we think that those companies that are linked with us are really quite interesting companies in themselves. Sometimes they're only very new companies, but they've got a really cool idea, which I think, or we think, can be brought in to help what we're doing. So that's the relationships. We haven't tried. We have actually sort of talked to the bigger companies, but there's a sort of at the present time, I don't think there's much happening in terms of those sort of deals going on at the moment. But um, you know, we've tried to want to continue doing it our way until we get to what we want, and then
Right.
give it over. As soon as you, as soon as you engage some of these big gorillas, of course, they own the space, and then they'll do what they want to do, and uh, and we want to continue to do what we're doing because it's a bit different to what other people are doing. Yeah.
So important. And I mean, this naturally leads me to my next question, which is, what sort of strategies and ways are you thinking about implementing scaling Cartherics as you go forward? And what sort of challenges do you foresee in the future?
There's a lot of challenges. There's technically challenges, financial challenges, there's regulatory challenges, there's all of them. We're meeting all of them. And I'd have to say, with each of these challenges, sometimes they look dreadful, they look awful, but it actually drives our team to even work harder, which is a bit of an interesting phenomenon. Um, so scaling is important. We're scaling the product and moving ourselves into bioreactors, and so you can go little bioreactors to medium to big. We're on that scale there now, and we can see that it is. You can actually move from one scale to another relatively easily, but you've got to keep making adjustments because it's a different environment with a bigger bioreactor than you had before. But we're actually now moving those cells into being able to produce very large numbers of them, natural killer cells anyway, and the T cells sort of make those reasonably well now too. So that scale is working all right. The scale of the company, we've just taken a reduction in our scale because of the financials to sort of keep our capital working longer so that we can get closer to our IND or clinical studies. But that hasn't hurt us. I think it just concentrated the effort again. And so the people have become really more engaged than they were before, perhaps. Well, they have actually, because they've taken on more things and even harder. So I think it depends what it is. When we meet a technical issue, we say, okay, who's actually, is there any example in a university or anywhere, a company that's actually been able to get around that? And then we'll either go talk to them or we talk to our academic friends and say, is there some way to get around this technical rock in the road? And it's very frequently that they'll say, yeah, let's try. And shortly thereafter, you've got an answer. So I think the scale up is one of where we want to keep working together with colleagues and not put too much cost into the company in terms of having a large entity, but keep our discovery going until we've got what we need.
Right.
And that's really crucial. We need to go to do clinical trials because the investment community won't believe you unless you're doing human studies, which is fair enough. But we don't think necessarily our first product will be the most ideal because we've got a lot more backing up behind that, which we think will do it even better. But if we get some readout, it'll certainly encourage the investment into the company and the support. So that's the way we're seeing our scale up. Don't rush at it. Get the science really tight down and working properly and getting to what we really want. Do enough of the clinical trials to keep our investors and those people believing in us, but make sure that you end up with something that will allow you to achieve your vision, which is what we really want to do.
Yeah. So, I mean, so absolutely critical, right? To take it step by step and piece by piece, make sure that each piece links as well. And yeah, I mean, it is so important. So there are 3 questions that I like to ask every guest. What inspires you?
Science has always inspired me. I think probably people would think I'm a bit of an opportunist to get into spaces where things suddenly are happening. But I think that's sometimes good luck. I mean, you have to be in a certain place at a certain time to engage. But it's really cool to do experiments that work. And it's not too bad even if they don't work, because it sort of drives you to sort of find out why it didn't work and then get around it. So being a scientist is a thing. I like going into the lab now. They don't trust me to do all the pipetting and things, but go and talk to them and then say, show me, oh, yeah, is that like that? Or, well, just show me. And, but I don't believe that looks as good as it was. Maybe, getting engaged with that, with the science is what drives me. I didn't do very well as a sort of occasional gardener for my wife. So she said, get yourself a job. So, I think retirement will come when the boss taps me on the shoulder and says, "Look, there's someone better," and that's fair enough because he's done it before. So that it's all right, you know. But if I can keep going and keep enjoying what I'm doing, and I think I can get to making a difference, then I'll wake up every day saying, "What's on today?"
Yeah.
I mean, maybe I got to talk to Don, but the next thing is there's something happened in the lab. I want to go down and see what was the outcome of that experiment. So it still drives me, and I think that's the thing. If you maintain the enthusiasm at the top, I think it can flow through to all the people. But if you have someone at the top that's not enthusiastic, it's a problem for everybody that to get that sort of buzz. So we open a lot of champagne bottles, because it's worth celebrating some experiments. And wow, we made 10 billion cells today. Or what, oh, we converted that cell to, did you do that really? Let's go and have a drink. I think that's what science is about, and that's what I've spent my life doing. And I don't know, people like you don't know what it's like, that buzz. It's pretty cool.
Yeah, I certainly do, know the excitement of crossing a threshold as well. In terms of, any research, especially whenever, it comes at a series of expenses, right? I mean, cells are not easy to grow. And, doing things and crafting things the way that you guys are is not necessarily an easy thing and should be celebrated as well. The next question for you is, what concerns you?
Hi, always it concerns me that that there'll be something that I can't govern that will interrupt what we're doing. Often that's, can you be sure that finances will sort of get you across the line? But so far we have such really good investors with us that it's kept us very, very well supported. So, but you can't always expect them to be there. There's a limit to what everybody can do. So you have to keep, looking forward. So the concerns for me is that, will I have enough time to get this vision established before something unpredictable happens that stops you? Um, that would be disappointing, I think, not to be able to not to be able to see some patients recover from ovarian cancer through the therapy. If they can do that, if we can start to see an impact in patients, I mean, that'll do me. But the concerns are really happen every day. Will the FDA give us permission? Will we get this and that done? You know, there's always concerns. But I think if you look at it in a positive light and respond to the experiment in a positive way, but also the FDA in a positive way, or whatever you're doing in a positive way, it'll generally get them to be more positive to you. If you feel negative about what you're concerned about, I think it sort of also, you know, doesn't help your case in whatever it is. So I've never thought that I'd be stopped in doing what I wanted to do, even though we had to go through a lot of political debate about some of those things in the IVF days and the stem cell days. They could have stopped all of that, but they didn't. We kept the people on side, the majority of the people on side, and I think we'll do it this time as well. And I think people will say, "This is why should they be successful when we haven't been?" Well, they didn't try this way, did they? So it's okay for people to be negative, but I'm enthusiastic about what we're seeing we can do. And I believe if you look deep enough at what you're doing and get the science of it right, you'll get an outcome that you expect.
Yeah, very good. Last question, what excites you?
I think the thing that excites me most is the science around us. Not only what we're doing, what other people are doing, and it's hard not to get excited about everything that's going on. It's amazing what people are doing, isn't it? It's just sort of amazing. And I mean, there's moments like Ian Wilmot telling me he made Dolly where we're having a hike up in the Edinburgh Hills and saying, it can't be right. Nobody's done that before. Everyone says it's not possible. And then I thought, well, I have to change my lecture notes, and that was a strange response. It was just an amazing moment. I mean, you know, those amazing moments happen, and if you're able to be there when they're happening, it's very special. And that's what I enjoyed about CERM. There were some very special moments there where, gosh, you made that happen. You know, you're able to get those cells to do that. It was just special. So science excites me, technology excites me. Some things don't, you know, there's a troublesome impediment about getting some really innovative stuff done. But then you have to accept that regulation and staying within boundaries is necessary. But there's a lot of things that we need to do to keep the people of this world healthy and the planet healthy. And I think science is a way that's going to help us do that, because I've lost hope in our political friends being able to do it.
Right. Yeah, as I sit here in the U.S. right now, it's not the greatest political environment that I sit in, for sure. And I certainly don't think, you know, they get anything done other than fighting amongst themselves. So for now,
pity, isn't it?
for now, we'll have to just set that one aside. That certainly doesn't excite me and is more on the concern side. So,
Right.
yeah, exactly.
Right. Yeah.
Well, Dr. Alan Trounson, thank you so much for being on the Life Science Success Podcast. I greatly appreciated our conversation and getting to hear about Cartherics and everything that you're working on there, and wish you all the best for the future.
Good on you, Don, and thanks very much for arranging this podcast. And Thanks for everybody who listened to it.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it. And last but not least, rate the podcast. Thank you again.